Good morning everyone, welcome back to another session. So body fluids and circulation from the NCERT. So if anyone is did not subscribe the channel, please do subscribe it. So we'll start the session. So what is the list chapter? Body fluids and circulation. So in this one small topic I am going to explain today, body fluids and circulation. So already introduction part we completed. If anyone did not watch that video, please do watch the video. The link is in the description. So here today's topic is blood grouping. Okay, blood grouping. So in the blood grouping topic, so in the world all people are having different types of blood groups. So what are these blood groups? So how we can determine the blood groups? So in detail I am going to explain in today's videos. See, so blood groups are mainly based on very important point here, presence and absence of antigens on the surface of the RBC. This is the main point. So we can determine the blood groups based on the presence and absence of antigens on the surface of the RBC. So here you have to know two words meanings. One is, what is antigen? Antigen ante enti? Antibody. So here antigen is responsible to induce the immune response, inhibit the immune response. Means it is not promoting the immune response. Immune response ni jarakunda aapedi antigen, immune response. Okay, so antibodies are present in the plasma cells. So antigens are present in the RBC, antibodies are present in the plasma cells. Okay, so these antibodies are promoting the immune response, which promote the immune response. This is the basic point in the blood grouping. Mainly we can determine the blood groups. Blood group ఇది ఏనా బీనా ఓనా అనేది డిటర్మైన్ చేయడానికి మెయిన్గా కావాల్సింది ఏంటంటే యాంటీజెన్స్. సో ఇఫ్ యాంటీజెన్ ఈజ్ ప్రెజెంట్ ఆన్ ది సర్ఫేస్ ఆఫ్ ది సెల్, if the antigen is present on the surface of the cell, that type of blood groups we can consider as particular type of blood groups. Like if it contains antigen A, the blood group will be A. Okay, next one, if the person contains the B antigen on its surface, the person contains the B blood group. And if the RBC does not contain any antigens, the blood group will be O. Okay, what are the types of blood groups now? A, B and O. So why they are divided into A, B, O blood groups? Because so in this ABO blood grouping process, A, B and then AB and then O. So here antigens will be antibodies and then blood group donors. Okay, blood group donors. So first one, the antigen will be A, antibody is B, and here antigen is B, antibody is A. So the blood group donor will be A and O, and here the blood group donor B and O. So here antigens both A, B are present, antibodies are absent, and here the blood group donors will be A and AB, AB and O. Okay, and then here O, so it does not contain any antigens and antigens both are present. So antibodies A and B are present in the O blood group. So and the donor is only O. So very simple blood grouping here based on the presence of antigens. So if the A blood group person, so A blood group person, definitely she should contain the A antigen, and here B contains B antigen, and AB contains both A and B antigens on the red blood cells surface. And then O, it does not contain any antigens, it contains only antibodies. It contains only antibodies. So that's why, so here we have very important 2 points. Here AB blood group considered as a universal recipient, means he can take any blood group person's blood. And then O blood group is considered as a universal donor, he can donate the blood to all the types of blood groups. So this is about the ABO blood grouping. Okay, so ABO blood grouping, once again fast quick recap. So we can able to determine the blood groups, presence and absence of the antigens and antibodies. So antigens are 2 types, A antigens and B antigens. So if A antigens are present, the person will contain the A blood group, B antigen, B blood group, both AB antigens, AB blood group, and if both antigens are absent, the person is O blood group. And now we will go to the RH grouping, RH blood grouping. So RH blood grouping here, RH blood grouping is first time RH factor, RH blood grouping topic. So RH blood grouping takes place after the discovery of the RH antigen in the RH rhesus monkey. RH antigen first time they observed in the rhesus monkey, that's why the factor is given with RH. So RH antigen, if the RH antigen is present, so this RH antigen is present, he considered as, if the RH antigen is present, he considered as RH positive person, and if RH antigen is not present, if it is absent, the person will be RH negative. RH negative. So RH factor it means RH antigen which is present in the blood cell surface, and if the RH positive person contains the RH antigen only, and if RH negative does not contains any antigens on its blood cell. Next one, here very important topic, that is if the person, if a child is having the different blood groups as different blood group persons as parents. So here we will see one circumstance. So simple ga entante, so mother, if mother is B positive and if father is B negative situations. So in the first child situation, the person will be safe. Why? Because the mother blood and fetal blood was clearly separated by the placenta in the situation of a first child. So in the situation of first child, the mother blood and fetus blood is separated by both are separated by the placenta. But in the condition of a second child, there is a chance of entering the mother blood into the fetal blood. Mother blood into the fetal blood results the agglutination. Means clotting takes place. Clotting జరిగినప్పుడు ఏమవుతుంది? So automatically clots form అయిపోయి the fetus is going to be deplete. So that's why if they know in the first condition only this is a situation, positive negative chances are there, though there is a chance of abortion in the second condition, better they avoid the second pregnancy. So very simple topic. This condition is known as second child condition. The second child condition, it results the problem as erythroblastosis fetalis. Erythroblastosis fetalis means the blood cells are going to be destroy the fetus because of the presence of the two different antigen antibody. It results the formation of the clots in the blood vessels and clots in the fetus, so it results the abortion. Okay, so that is all about the blood grouping. Okay, I hope you all understand the session. Please like, share and subscribe the channel. Okay, thanks for watching.